So hi, my name is Alex Cope, and I am a PhD candidate in the Graduate School of Genome Science and Technology here at the University of Tennessee, Knoxville. I'm currently studying molecular evolution, which just means that I think about how has a gene evolved, what evolutionary forces are shaping these genes. And a natural question that extends from that is, why did a gene evolve in this way when we see that these other genes evolved in a different way? So more specifically, I tend to view molecular evolution through this kind of cost-benefit framework. A cell is obviously finite. It only has so many resources to dedicate to all the processes that it needs in order to survive. So you might have a mutation that changes the gene, which then changes the cost. And it might or might not impact the benefit to the cell. Regardless of whether or not it changes that benefit, if the benefit does not outweigh the cost, then there's a lower chance that that change is going to become fixed in the population, or the most common version of the gene, just because, well, the cells that have that mutation are less fit, they're less likely to reproduce. And obviously the opposite is true. If the benefit outweighs the cost, now we're going to see that it's more likely that that mutation is going to become the dominant version of the gene within the population. So defining the cost-benefit trade-off obviously is going to vary depending on the biological context and the question you're trying to ask. For example, let's take creating a protein. You can, in this case, your cost function could be something as simple as, well, the materials we're going to use to build the protein, or even thinking about the cost to the cell and changing the assembly speed of the protein. In fact, the latter question is relevant to a lot of the research I've done right now, thinking about, well, how do changes at the nucleotide level affect the assembly speed of proteins in relation to protein secretion and protein structure? Okay, so to actually address these questions in molecular evolution, I employ mathematical models that use this cost-benefit framework, contextualized also within a population genetics theory. And population genetics is just thinking about how do gene frequencies vary within a population. By combining these two elements, I'm able to distinguish between what are the selective forces shaping a gene versus what might be due to non-selective forces, such as random biases in the mutational process. So ultimately, being able to identify and understand the selective pressures shaping a gene can provide insight into the mechanism in which it functions in the cell. And that in turn can lead to insight into how this gene functions to shape the traits an organism exhibits.